So as in the first session or in the first webinar, you already discussed like what is sequencing, how we are going to be processed with the sequencing, right? But DNA sequencing, actually the aim is not just to know the ATGC here. The aim of the DNA sequencing is what, like whenever you wanted to find out, like whatever the sample you are taking, in that sample, is there any changes you are getting? Is there any variations you are getting? Variations as in you are getting any SNPs or indels within that genome or within that exome. So at that time, you can go with the DNA sequencing. SNPs as in what, like a single nucleotide polymorphism, means when our codon, if any base is got polymorphited, so that's what we call it as a single nucleotide polymorphism, which may leads to change your amino acid or may not. Okay, depend upon the type of SNP. Also, if you are getting any insertion deletion related with that, so if you wanted to find out, you can go with the DNA sequencing. But actually, how you are going to identify it? So as you can go with the reference genome or a exome sequencing. It's like if you are taking a whole genome and you are aligning your data with the reference genome, that time you can able to identify it. Means here, whenever you are getting a raw data from a sequencing company, like if you are having any sample, you are interested, you are doing a sequencing by preparing the library, and you are getting a raw data in the form of short fragments, right? So you can take a reference genome of that organism of whatever the data you are taking. You can download the reference genome from any one of the database in which you are interested, like NCBI, UCSC, Ensemble, or whatever it is. So once you know the reference genome, you know in the reference genome already the bases we are having. So with the help of some tools, those fragments or those beads which you are getting from the sequencing company, you can align. Okay. So once the fragments are aligned, then again, with the help of some tools, we are going to match those fragments to the reference genome. So you can see this diagram. If let's consider in a reference genome, if any base is present, and whatever the fragments they are aligning here at this position, it is showing some another base. So that thing we are going to detect as a mutation. Okay. So basically DNA-seq pipeline, it's going to be identify the variant, you can say a SNP, indel, within the whole genome and whole exome sequencing. Whole exome sequencing is what it is going to be identify the variation in the protein coding genes. Because as in this region, most of the large variations or large SNPs we can get. Because protein coding region is a 1% of the whole genome. So if you are like you are only interested with the protein coding region instead of the whole genome, so at that time, am I audible?
Yes, ma'am, you're audible. I'll also request you to please put your presentation in the presentation mode.
Yeah, it's okay. Fine. So yeah, like actually there is a protein coding we are having as a 1% of the whole genome. So if you're interested with the protein coding genes only, you wanted to identify the variations in that, you can go with the whole exome sequencing, so that the like size of the data will be the small, so your sequencing cost will be also low at that time. So it's depend upon your aim and your choice. So actually, why the SNPs are important? Like why identifying the SNPs or like a variation is important in our why it is important? Because actually the SNPs can be used as a biological markers, means like used to identify, to locate the genes which are associated with a particular disease. And whenever you are getting any SNPs which is occurring in any particular region or a region of gene or any regulatory region, so they play a more direct role in a disease by affecting the gene function. So that's why if you are dealing with that, you can go with the DNA-seq analysis.